Okay, so I think we are ready to start. Welcome, everyone. Welcome to Neurologics webinar. I'm Rafał Nowak, and I will be leading this webinar. And today we'll be talking about neurourbanism and emotional cities with scientific evidence on how the natural and built urban environment shapes the neural system underlying human cognitive and emotional processing. And this with a perspective that incorporates age, gender, and vulnerable groups. And I'm very happy to welcome our guest speakers. We have today with us Dr. Bruno Miranda and João Amaro. Hello, welcome guys, and thanks for joining us.
Hello, thank you, Rafał. Very nice. Thank you for the invitation.
Hello, thank you.
Thank you very much. So just a little housekeeping before we get started. So we'll begin with a presentation, and this will be followed by a Q&A. So please feel free to submit your questions at any time using the chat tool. And for information, the recorded version of this webinar also will be available offline. So please let me quickly introduce our speakers. Dr. Bruno Miranda is a neurologist and assistant professor at the University of Lisbon, specializing in cognition, neurodegenerative diseases, and the impact of environmental factors on brain function. He holds a PhD in neuroscience from the University College London, where his research focuses on computational models of decision-making. His current work explores the interactions of neuroscience with urban planning and architecture, using methods such as behavioral analysis, digital health, neuroimaging, and computational modeling. Dr. Miranda also co-leads the Emotional Cities Horizon 2020 project with studies that influence urban environment on health and emotional well-being. João Amaro is a master's student in neuroscience at the University of Lisbon, works with an emotional cities project focusing on EEG-based analysis of cognitive and emotional responses to different urban settings. And his research integrates EEG data with behavioral, physiological, and environmental measurements to explore how real-world environment affect brain dynamics. So together, our guests combine expertise in neuroscience, urban design, and technology to better understand the brain's interaction with the urban environment. So thank you, guys. The stage is yours.
So thank you all. As Rafał said, we both from the University of Lisbon, the Lisbon School of Medicine. So I will outline here what the presentation is going to be about. So there's this first part where I will introduce and give a little bit of the background of the Emotional Cities project, going into a little bit of the details of our experiments, not going too far, but just highlighting the ones that are a little bit more relevant for the type of audience that we were expecting. Also because our emphasis was a little bit more on our humbled experience of mobile EEG. So we have to say that we are not as experienced as other groups in mobile EEG. This was, as you will see, part of the project that we felt could complement the work that we were doing with other experiments. And so I will handle at the second part for João Amaro for him to share the hands-on experience, because that's what also I think the webinar might be helpful for people interested in the field, because he was the one actually running the experiments in the wild. And so I think it will be interesting to see some of his comments. So just a quick introduction for the topic of the emotional cities. We are becoming urban species. So as urban population is growing, there's a lot of challenges. One of them is actually mental health problems, which is some of the major priorities in global health. And it is known that urban living has been associated with mood-related or anxiety-related disorders. I'm not going that far into the other type of psychiatric, but there's also evidence suggesting that there's an increase of other more specific cases of psychiatry. But if we think about what actually determines our health, either physical or mental, although I have to say that in our case, we're a little bit more biased towards the mental perspective. One can argue that we have individual characteristics from, on one hand, the more biological perspective. But if we look at the urban literature or the urban studies, as I've been contacting with the field, so I'm also new to the field in a sense, there's also evidence of people in the urban studies that have been focusing their work on making cities that are helpful for people and looking at these sort of individual elements or individual characteristics. There's also an environmental context. In medicine, we always say about the genetic and environmental factors, for example. And also in the literature of urban studies, we can find examples of some authors suggesting that actually anything about the vibrancy of the city, of the social element is also very important. And finally, we are the ones that also make our decision of doing some particular behavior that we are aware that or not that may cause some health issue. But again, is our decision. And also there's in the urban literature this notion that the way you experience the city is also important, like that you also have to conceive the city as a way to guide actions or help these decisions. And so if from my perspective, because I'm not born in the urban studies field, so I was born in the medical and neuroscience field, the way I look at this from the perspective of neuroscience, which combines a lot of various approaches, is something related with what I did in my early PhD studies, looking at how an agent interacts with the environment. And this is part of the reinforcement learning theory, one of the most contemporary learning theories with a lot of evidence from animal, human evidence that just relates how the agent acts and interacts with the environment through this computational framework, and how we achieve reward. And this reward could be seen as a sort of feeling well or feeling this notion of well-being. But of course that the environment changes across time through states. And we have the possibility of acting in this interaction. So a new discipline that some have suggested is the idea of neurourbanism. On one hand, it's also fancy to put the neuro on top of some other words, but I think this idea of bringing different type of evidence to this field of how we understand this human environment interaction is actually important. And so that makes sense from the perspective of what I just introduced. That's where I think the emotional cities was born, from a theoretical conceptual framework. But the emotional cities is actually a Horizon 2020 project. I also highlight that we are part of the urban health cluster, which reunites several other projects from an urban health call of the European Commission. And the project will try to provide evidence on how the built urban environment influence our mental health. And we will do this by tackling a little bit some key topics, particular vulnerable groups. In our case, we are not presenting here data, but we selected in one experiment the mild cognitive impairment group. And we also will look at some differences between age, although this is hard sometimes with the sample size to tackle this adequately. But our idea was to create a infrastructure to hold the data as well as collect the data, to combine these different data sets from the different perspectives. So from the neurological perspective to the urban perspective, how we bring together some different levels of human level data, group data, big data, and things like this. And at the end, for our funding agency, we also have to provide something about policy recommendations. And sometimes it's hard to go from data to policy, and that's what we are also facing. But the consortium we have is 12 participants, a good equilibrium between academia and industry. We also have a partner from outside Europe, and as the call highlighted, that there was this need to interact with another type of experience outside Europe. It's a project that is about to finish, in a sense. It's February, we'll be finishing the project. So it was a 48-month project. There's several partners involved. So the Institute of Geography and Spatial Planning from the University of Lisbon is the is the official coordinator, but we are co-coordinating this because they are more from the field of the urban studies. We are more from the neuroscience. And so on this image here, we have this on the top, more like the academic perspective, and on the bottom, the industry perspective, small, medium enterprises. And on the left, you will have more focus on the urban elements, and on the right, something to do more with biology or neuroscience. Here, a little bit of the dissemination and communication group that we have. There's a little bit here in the middle, but we do have several different approaches. So we have something to do more with microclimate, with mobility and land use. Also, with the Michigan State University, we developed this fMRI study. We also have the contribution from StarLab, Tartu with psychology, StarLab with the sort of EEG data analysis support for the use of the device that we add provide EEG, the neuroelectric Zenobio that we will be presenting. The integration with the NeuroGears, the group that has a part of it in UK and also in Portugal, was also important to bring this together on a wearable kit. Byte was the one responsible for merging a little bit the data from the big data from the urban together with the NeuroGears to support in the inclusion of data from the neurological aspect and climate flux, addressing some microclimate issues. So our approach started with what neuroscience can provide for urbanism. So of course we had to think about some topics, and topics like emotion, decision-making, spatial navigation, or social behavior are critical in neuroscience. But we had to adopt like a lab-based first approach, using the technology that is conventionally used in neuroscience experiments to get some data to guide us for any outdoor experience that we might want. And of course, if we go to the outdoor, we will need wearable devices, and this is technologically a challenge to also combine this with, for example, environmental sensors, because if we go to the outside world, we also better take that into account. And we also have these other tools like apps that can help monitoring. We selected 4 case study cities: Lisbon, Lansing, London, and Copenhagen, because our partners were also there and they had different configurations or morphology. But finally, if we want to also tackle urban health, we would like to have a population. And as I said, we selected a mild cognitive impaired population, and we conceived like a spatial cognition task to see virtually with modifications whether this can impact these patients. So, but this was a generic approach. We had to make these actual experiments so that we can test hypotheses. And so we conceived these 5 experiments. One was a little bit more indoor with fMRI, and that's a study that with the first outcome has been already published in the Nature Cities, just recently been released. 2 studies, the 2 experiments I will talk about are experiment 2 and 4, and then there will be experiment 3 and 5 that I wouldn't have time here. So I will be focusing on experiment 2 and 4. You can follow the data, the findings from the Nature Cities about experiment 1. And I think we decided this because it's the appropriate experiments to explain here. So what was our goal? We wanted to see to run some experiments in the outdoor environment, to start with this sort of mobile imaging that others have already started years back, but we had to make some decisions. So we started basically by addressing some neighborhoods and with the spatial data analysis first to guide where to run our experiments. And so to get to an individual and granular element, we selected some walks. And so for just to give you an example of Lisbon, one of our cases, we had some various places that we thought we could run some experiments. By the end, we ran in these sort of 6 places because we had to compromise for the data sample size that we were going to collect. So if we are on a city walk, course, that 20 minutes of a walk or 15-minute walk that conventionally now is becoming a trend in terms of cities, that's what we selected. We cannot just run experiments in the lab, 15 minutes walking. We could, but it could be hard to get people motivated. But we selected some small parts, and João will explain a little bit more. We ask about how people feel in terms of emotion from a qualitative perspective or valence, or from a quantity perspective, the arousal, and we recorded. The idea was with some help of the statistics and the modeling to see how we can map cities according to these sort of emotional reactions. So the idea with the first experiment in the lab was to look at some stimulus, and we selected videos to show and just ask them 2 questions about valence and arousal and see what the responses of people. And so I'll show you just one example of a video. Here is just like this 20-second video that we present. And the importance here is that we, together with the urban team, in this case, the eGOT from Lisbon, for example, we actually track where these walks were, and we could now start making relationships not only with different type of data that urban studies usually gather and play with about land use, about space syntax and things like this, but we also are trying to work with computer vision segmentation to help, for example, studying other more particular elements, such as how you relate with people and people's presence. We also conceive like more psychological perspective of self-reporting. And in this case, for example, this video was actually classified as being very natural, in a very natural environment and with not many people in the video, which makes sense. So they were categorized on these 2 domains because we felt these were domains that we were interested. And then we applied these methodologies to extract features from the brain, from the behavior, from the physiology, with wearable devices that we were also using in the outdoor, but with the EEG in this case, with a high density EEG in the lab, and also with eye tracking. And so you could see here just some patterns of heat mapping of where people are looking at. And so the idea, though, is that if we want to apply this from experiment 2 for experiment 4, which is the one outdoor, we would say that, okay, we are close to we need a design that is similar, because we are jumping a lot of hurdles and the path is not really linear. There's a lot of issues. And for example, the work by groups in mobile EEG actually highlight these issues of how you can be sure or perform this rigorous science. So for that, we developed some protocol that could help liaising the indoor findings with the outdoor so that we can test hypotheses and not be completely in an explorative sense in this outdoor perspective. And João might explain a little bit more these, but we collected, for example, in Lisbon, 60 acquisitions, 60 participants. Some of them actually run 3 different paths in the ones that I've shown, but majority only ran 1 path because logistically this was complex as well, given also the time frame we had. And we tried to include a little bit of various age groups, but of course some of the easier ones to recruit were the younger ones. And so this is an example of a walk that we did in Lisbon. And we have a starting point and then an end point. And we have some checkpoints that actually relate with the video. And so these are points where the videos were performed, and then we can make the link between the video and the outdoor. The interesting part was this tool that we had to develop for the wearable kit to be a sort of mobile EEG type, but we also included this microclimate station together with Climate Flux partner, and we also integrated and synchronized this by the work with NeuroGears, synchronizing all these devices. We also used eye tracking and this is how it looks like to walk. We use Empatica for the wristband, and we will soon try to publish like the framework of this data synchronization. But as I said, this was a lot of the work done as well by the NeuroGears team, where they actually put together these wearable kits so that we could have the signals, as João will show, synchronized. And we know that, for example, in this red path, we can have the sense of the temperature, the altitude, the speed the person was walking, heart rate, and then we could add as well eye tracking and also physiological. So there's a lot of work by, for example, eye tracking. Leonardo, Marta is doing physiology, Pedro is doing clinical, Diego was helping us with the functional imaging study. And of course, a lot of this is in collaboration with, at least in Lisbon, the data that I'm shown, with the group from the IGOT, Paulo Morgado, the other co-coordinator, and then Rafael, who helped as well a lot in the data collection. And of course, our partners, as I said, that at least are related with some of these, and that also are working Star Lab in the EEG analysis. So I think João is going to show you a little bit the hands-on experience by using the wearable kit and the eNOBIO 32-channel device that we used. And so I will leave now for João to show a little bit some of his findings or challenges and what we had. So the idea, unfortunately, is not to share a lot of data, because we are in the process now of analyzing collected data. And so we were not comfortable yet of showing a lot of data. And I feel that it's important as well to share some of our hurdles and obstacles that we faced, or some highlight that you might conceive in this field.
Okay, now, as Dr. Bruno said, I will go into more of the details related to actually doing this outdoor mobile EEG study. And I'm going to start from acquisition to inspecting the data, and then to the analysis part. So starting with data acquisition. So I will start with the in-field perspective, but first I will give a quick recap of our actual paradigm. So as Dr. Bruno already said, we had two very similar paradigms. We have an indoor EEG study where the person is seated down and watching a succession of videos, which are taking around several areas in the city of Lisbon. And then we also have participants doing the outdoor experiment, going through walks, which contain some of the videos which were shown in the indoor experiment. And we tried as best as we could to make the paradigms comparable. And we did so by, for example, in the indoor experiment, each video was preceded by a 1 second pause of the first frame of the video. So we could assess activity related to more unconscious procedures in the brain. And the equivalent of this in the outdoor experiments was asking the participant to stop in the place corresponding to this first frame of the video, and telling him that he could contemplate his surroundings, of course trying to minimize movement. Then in the indoor, the video actually started. It was a 20-second video, and the outdoor experiment, we tried to replicate as much this 20-second walk as well. As we'll see later, behind the participants, with the researchers are giving instructions for the participant. And then in the indoor, we also have questionnaires about the valence and arousal. So valence is about how much the participants like the video on a scale of 1 to 9. And arousal, how strong did those emotions. For example, a 9 in arousal would be the person feeling very active, while a 1 would be more calm and relaxed. And these same questionnaires were provided to the participants in the outdoor experiment with a tablet that we gave to the participants. And because the outdoor walks just had a few spots corresponding to the videos, then each path had a different number of checkpoints, which correspond to this workflow of events. I also like to point out that each participant, they filled the same personality questionnaires, which can later be used to get a more characterization of the participants themselves. But before we go more into the data and the technology, I will start by pointing out the logistics side and what we actually need to do to actually do this experiment. So we used to take the backpack in a travel bag. So luckily our team did a good job in making sure that the backpack was small enough to be carried around. And usually we arrived at the starting point and we take the backpack off the travel bag and we start preparing the sensors, all the external sensors, which will later be connected to the backpack. And here's an image depicting that moment. Then we give the subject a questionnaire to assess his momentary state, because even though I already referred that we have personality information, we want to have more momentary information about how the person is feeling at that moment. And then finally, the last step is actually putting the backpack on the participant and giving him instructions. Usually the instructions consisted of, the goal of the instructions were to be able to make the participant as comfortable as possible and tell him to abstract away from all the looks that inevitably are going to be present from other people. And of course, the weight that the backpack also gives. We tried to ask the participant to try to imagine it was just a casual walk. So for this experiment, we decided to go with eNOVIO32 system. This system provided several advantages for the outdoor setting. For example, we have 32 channels, which is a good spatial coverage of the scalp. This system provided an easy and quick setup, which is essential in outdoor setting, and especially when we have a lot of participants scheduled for that day. We cannot afford to lose a lot of time. And for example, on the image on the top right, we show one researcher putting the stickers with conductive gel, which will be used for the reference electrodes. So it's very easy, takes 5 seconds to put it on the participant. And on the bottom right, we show the electrodes, which need to be fitted into the cap. And these are dry electrodes, so they do not need gel, which facilitates a lot with the procedure, making it a lot faster and quicker. Another advantages of using eNOVIO, for example, was a good acquisition software. For example, it provided real-time filtering of line noise, and it also allowed from different modes of acquisition. For example, we acquired EEG data by cable, as you can see in the image at the center, but it would also be possible for Wi-Fi. Some limitations that we found were that these electrodes were a bit uncomfortable, and we did walks of around 25 minutes, and by the end, it started stressing a bit our participants. And another limitation was unoptimized cable placement, connecting the amplifier with our actual backpack. So unfortunately, due to the way the connection was placed, there was a lot of pressure being made on the cable because it was in contact with the neck. And this sometimes resulted in actual uncomfort for the participant. And so if it was possible, it would be really amazing. We'd like to suggest that this, for example, this cable could have been placed on the side, which would really help a lot. Okay. But moving forward, so here on the top, we have a scheme depicting the path itself. A represents the beginning of the path and B the end. The stars represent the checkpoints, and the colors represent different typologies. So this is how we represent our paths for later analysis and characterizing the paths. And basically the participants would go from A to B, with the researchers going behind the participants, always giving instructions and getting real-time feedback from the backpack. So now I will show you what we're actually seeing on the backpack. This is the output of the underlying programming language that synchronizes all data, which is Bonsai, also developed by NeuroGears. And this is our graphical user interface that gives us real-time feedback of the sensors. Here, as you can see, on the top right, we have eye tracking data, real-time. On the bottom, we have ECG, and everything seems to be going fine. I also want to point out that here on the left side is the EEG. These numbers correspond to event codes, because while the participant is doing the task, we're also inputting the event time and what it means. And below it are all the sensors which the backpack is synchronizing. And of course, the red one means not working, although this specific sensor refers to starting points and always turns red when the participant starts walking. But here we actually have an example of actual sensor failing, where our eye tracking device stopped working, and so it turned red. So unfortunately, we had a lot of cases of missing data, of incomplete and absent data, and it is also expected due to complexity of the backpack itself. For example, in the case of EEG, the most common cause of failure was, as already mentioned, the cable connections. So either because of a lot of use, the ports for the connection started getting loose, or the cables would disconnect because of some abrupt movement. In the case of Puppy Labs, which is our eye tracking device, and Empatica, Empatica operates via Bluetooth and Puppy Labs via Wi-Fi. And unfortunately, we're using mobile Wi-Fi. And if just at any instance this Wi-Fi connection failed, then we would lose Puppy Labs from that moment on, which also explains its high, almost 50% rate of failure. Then in the ECG, our most common cause of failure was basically not using good stickers, which contain the conductive gel. In the case of the microphone, it was actually the batteries. They were out. In the case of GPS, we have low failure because this only happened in places where the top of the backpack, so the backpack got GPS data from satellite, and if there was ceiling or high buildings nearby, this GPS data would be blocked. But this only happened on 2 paths at most. Regarding accelerometer and climate data, it was most common cause of failure was sensor failure, which is more technical. So in summary, the main motivations for doing outdoor mobile EEG are the fact that in the lab, the participant behavior is very constrained. The lab doesn't use naturalistic stimuli as being in the outdoor environment itself. And lab studies also lack environmental factors that can affect the actual data, such as temperature, humidity, and so on. So now I would like to compare the system we use, the EEG system, with what the literature recommends as what should be done in mobile EEG. So indeed, we have used eye sampling rate. We also use secure electrodes as they are attached to the cap, so they don't move much, and they are in a standard 10-10 system. We also have motion sensors, which can afterwards be used to give us a better sense of where we have most movement artifacts. We have a lightweight EEG system, so it was very easy to carry on the InnoB32. And we also use an open source approach, as all this procedure of synchronizing data at the software level and combining all the hardware into a backpack is all freely available. Then I would like to point out that although we have good electrode density, we have 32 channels, it is still better to use 64 or more channels, as is recommended in literature. So there is an advantage, however, of using 32 channels, which is, at least it is said in literature, that above 20 channels, you can get an effective ICA decomposition, which is essential for data cleaning afterwards. But we have also some limitations or things which are not as good as the literature tries to advise, which is we use a mastoid reference. So as you might remember from the last slides, I showed the image of a researcher putting the stickers on the mastoid bone behind the ear. But unfortunately, there are limitations. As people with more hair, it becomes more difficult to get better contact with the scalp. And people have different bone typologies, which also makes it difficult to always get an optimal position. And unfortunately, the reference is always the most important part of any EEG system. If you do not have a good reference, you're not going to have good data. Another limitation was the cable sway. when the participant was walking, unfortunately, the amplifier was moving a lot as well, which gives artifacts. And the fact that those dry electrodes, they are a bit uncomfortable, as I already said. And the last part, which we can also improve a tiny bit, is the ergonomics of the backpack itself. So the backpack weighed around 9 kilos, which when you put it on, you don't feel it as much. But after 25 minutes, you start feeling it. And also due to the climate sensor going upwards, the center of gravity is also pushed a bit backwards. Okay, so now I would like to go into the part of inspecting the data. So here, as I already said, we have two connected paradigms, and here I want to show you actual video data from the left of a participant doing the outdoor experiment, and on the right of a video shown in the indoor experiment. So as we can see, it's the same area. There's a lot more movement from the subject as he's looking around as expected. But overall, I think we succeeded in making the participants be on the same stimuli. Then I just want to show you what our output is like. Here we have different climate sensors, and it's basically showcasing how climate data can be synchronized at both the spatial and temporal levels. Now, going on to the EEG part. So the most obvious disadvantage of doing outdoor EEG is its vulnerability to artifacts. And this is going to be especially true for muscle activity as the participant is walking. So here is a EEG voltage in a 50-second, around 50-second time period. The 35001 is a code event that denotes beginning of walking. So we say start walking, the participant walks, and immediately we get these artifacts. So this needs to be accounted for later in the pre-processing. Not only is the data prone to movement artifacts, but also I show here a case of where we started in Copenhagen, an acquisition near to a train station. And the line noise was so strong that even with a real-time NOT filter applied, still we get a lot of power at 50 Hertz, which is depicted here in these high-frequency bursts in the time domain. And finally, I just want to showcase a graph whose purpose is basically to show the difference between EEG systems in the indoor lab and the outdoor lab. So this is data from one participant, the same participant that did the experiment outdoor and indoor. And in both cases, the participant was doing a baseline task of eyes open. So, of course, assuming if we think that the scenarios are similar, then we would expect this plot to be similar. But what we see is this variability in the GFP, which is global field power, which is simply a standard deviation of the data. This basically showcases that outdoor EEG is susceptible to external sources which we cannot control. For example, this data in specific, the participant was doing the baseline task in a very crowded area with cars going by, people walking, which might explain these bursts. Also, in the outdoor setting, the participant is standing, while in the indoor one, he's sitting down. Okay, so now I'm going to talk a bit about analyzing this data. So the most important part is actually how to pre-process the data. And we made sure that we search literature for the best standards and recommendations. And our goal here is to apply the same pre-processing pipeline to both indoor and outdoor data. So because in the indoor data, we use a different system, very high density system with 256 EEGs, the first step was to make sure that an equal number of channels. So we ignored every channel that did not correspond to the 1010 system used in the outdoor setting. Then we downsampled the data to 250 Hertz just to save space, and we removed bad channels. These bad channels were basically channels with no data at all, zeroed out channels, or channels without good correlation with neighboring channels. So now in orange are the actual artifact rejection procedures we implemented. First, we applied high-pass filter at 2 Hz, which might be high, and it is indeed compared to indoor EEG preprocessing. But as we looked around, we saw that 2 Hz high-pass filter improves a lot ICA decomposition. So we went with it. And for the indoor data, we also applied the notch filter, as it did not provide real-time line noise cancellation. Then we actually performed independent component analysis and rejected it with IC label from EEG lab. Then we also low-pass filter at 40 Hz to remove high-frequency bursts, and double down on this approach with burst rejection with ASR. Finally, we interpolate those channels and average to a common average reference. So as of now, we just finished data acquisition and the preprocessing pipeline that I just shown, and we have some future perspectives to analyze the data. So our first approach will be to assess frequency features, mainly frontal. For example, the frontal midline theta has been associated with cognitive controls, indicates heightened cognitive engagement or concentration. Therefore, it can be used to gauge the level of mental effort required in different environments, for example, reflecting how design elements impact cognitive engagement and fatigue. Then frontal alpha power is also associated with attention restoration, relaxation, and inhibitory control. So its correlation with relaxation or stress in response to environmental stimuli can be used to better understand how different settings promote or hinder mental well-being. Then we also have this theta beta ratio. It has been implicated in cognitive load, attention, and ADHD. Higher values correlate with mind wandering, and low values indicate more attention. And this seems adequate to test the effect of urban typology on our attentional resources. And finally, also frontal alpha asymmetry. Basically, in the literature, it has been described that negative values of this feature correspond to negative valence states, and it can be used to represent the positive affect and motivational approach of the participant in relation to various urban stimuli. So this is our approach.
Yeah, great, João, thanks. I was just going to say that we were in a way trying to have a conclusion slide, but we were struggling to have some conclusions, because the conclusions will be when we actually have our data analyzed. But I think we at least can say that we did our best to acquire data that could be from a perspective of what our objectives were, to provide as much scientific evidence for some guidance on how to help or use neuroscience to help urban planning. Sorry, I won't take more time. So Rafael, thanks for inviting.
Thank you very much. Congratulations on this project, very ambitious, and we are expecting for the presentation of the results, obviously. So like we are ready to take questions in the chat. So please use Q&A tool for the questions. I have actually one more technical, and thank you for all the feedback that you provided. And regarding the synchronization, in terms of different devices you are using, were they synchronized or they were somehow separately recordings? I mean, EEG, for example, with eye tracking, with wearable, or they were independently recording, or there was synchronization between these devices, or camera?
No, as you saw, everything was, so this was actually a part that was quite challenging, and we appreciated all the effort that NeuroGears helped us. It was actually the part that was hard, but João, you maybe can put there the video where so actually everything is in real time synchronized. So even the eye tracking data, yeah, that one. So everything here is actually synchronized. Yeah, so everything here is going through the same platform, okay. And so it will be everything aligned, so that you can also build the graphs that I show you, where you can just on the same time you can know all the variables at the same time. Of course, dealing with different products, of course, the intellectual properties of each device was something that we had to deal with. but we also try as much as possible to use for example, the LSL protocol for the EEG, and the Empatica. We also struggled initially to get these streamed for the pipeline that we were developing, and everything is also synchronized with environmental sensors, so they are actually connected.
Okay, perfect. Thank you very much. And I know that you will be still analyzing the data, but from the big picture, what would be the environmental features, and especially if you are talking in neurodegenerative disorders, for example, in MCI or Alzheimer, that could somehow mitigate the sensory challenges in this type of patients, or reduce the cognitive load that we know that MCI and Alzheimer's patients are struggling with. Do you think about like specific features that could be improved, or you will get based on your studies that will be provided as a feedback for the planners of the urban environment?
Yeah, thanks. It's a good question. We cannot provide a direct answer to that. So I do think that is a big gap between showing some evidence of, for example, cognitive load in certain urban environments, to say that this is actually what's causing problems with neurodegenerative disorders or some other type of cognitive disorders or some other type of health group or clinical group. It's a big step. What we conceived in the emotional cities was, again, although it looks ambitious, but we had some things in mind, that if we know that this is a particular environment that might be cognitive demanding, then we identify these features from the urban perspective or the environmental perspective. And we do actually have an experiment with the patients where some of these features are actually tested. So it will be an indirect relationship, saying that, for example, some urban features are associated with cognitive load from some of these studies that we've just shown. And on the experiment 5 with the clinical populations, we will say, actually, there's a decrease in performance of these patients compared to controls. And therefore, we expect that this might be because of a cognitive load. So then we will have some evidence from these other experiments saying that indeed the urban elements are the same that we are testing on this experiment 5. So then we can make these step with intermediate steps, so that we don't jump for final conclusions that might not be true, in the sense of getting the right answer.
Okay.
So
okay. This is sorry.
We will integrate this. So so I think by the end of the day, it's when we actually can combine a lot of these other experiments together that we can start building some of these assumptions. But I think the next step of some of these studies that should move forward is actually test this on particular settings, or on some specific settings, or particular populations, or things like this.
Perfect. And I understand that there are different working packages within the project that are studying and checking, researching the same from the different point of view. There is a question from Christina, actually, regarding another technique that is FNIRS. So Christina is asking this, like, for the future projects, if it would be possible to add FNIRS portable device to give us actually additional modality with near spectroscopy recording and additional to the EEG signal. What would be the limitation, and what would be the pro? And if you are thinking about using also FNIRS together with EEG, for example, for this type of recordings?
Yeah, it's a good question. I can maybe I can answer, João, but maybe you can also comment if you want. But yes, we are aware that there's a recent new evidence suggesting that this could be a potentially interesting technique also to contemplate for these outdoor settings. I think from my perspective, I haven't tried yet, so I haven't played with it. From what I've seen in publications, is mostly for frontal areas that has been reflecting interesting signals. I think there's also some technological advances with the technique that can expand to other areas of the brain. I think it will be on our setting, as long as it can follow some of the similar for example, LSL protocol, and depending on the device type of openness in terms of sharing the raw data and things like this, I don't think it would be too hard to implement this extra. And I think advantages can be a little bit of better localization and relationship with brain fMRI studies as well. That could also be very insightful.
Yeah, in the simple way, it would be like taking EEG out of the lab and taking fMRI out of the lab. Obviously, you cannot directly compare fNIRS and fMRI, but you have these different measurements on the neuronal level, electrical, and also oxygenation level.
There was been as well,
yeah,
just one more final thing. There was as well, like from as far as I remember, for example, someone that we interacted with as well, Arnold Wilkins from Cambridge. I think he's been also working with visual comfort and the perceptual element, and he's actually shown this signal as well with this type of technique that correlates well with the signal that they see from more posterior. So it's, although I was focusing more on the frontal, but I'm sure that this could also be applied to other areas, but there's a little bit of limitations yet with the technique.
So actually, next question is regarding the next 10 years, how you envision the research in the forthcoming years, and especially with improvement in the portable devices, and what kind of additional tests could be added to optimize the setup and the recording. And actually, follow-up question from Jim would be, what kind of treatment could be applied when you have when you identify these biomarkers, these changes on the specific factors? I think that this is like very going into the future, but maybe you can provide some clue on this.
Yeah, I was gonna say that so that I think we are just starting a little bit, so I think we might get really excited, but there's like the risk of going into the field of doing a lot of this without a lot of caution about the data that we're collecting. So I think there's going to be a need in the next 10 years to, on one hand, improve when do we need to go to the outdoor, because otherwise we just be spending a lot of time. So I think a lot of people will jump straight to do all these explorative studies in the wild. But if we lack robustness from the scientific perspective, we might find conclusions that are not necessarily relevant. But I also feel that the important part is also to think about what do we want from this data and how we can combine this with other types of data. And that's also what we are trying in the emotional cities, is to relate this with other type of data, this individual data with other type of data, and see where this actually helps. And so in terms of treatment, so first of all, I think if we try to prove that there's something that affects, or there's some parts of urban design that for sure, rather than just an expert view or an opinion view, that actually show some performance difference in terms of even some activities of daily living. And in our case, we adopting spatial cognition, in a sense that these people, mild cognitive impaired, they are still autonomous in their lifestyles routine. And so if we improve a little bit, they might have a bit of a deficit in navigation, but if we improve, and if we know how to improve with some data, that's already a treatment. It's almost like a prescription, but is a prescription that who's actually signing the prescription are the policymakers or the city counselors that are actually doing this prescription. But that's how I would see. One can also speculate that we may treat some patients with some interventional, like stimulation protocols to see if they can feel better in a particular environment. But I think that is going beyond what we currently have in terms of knowledge of the field.
I would also like to point out regarding the question of where design of research is going to be in the next 10 years, that with this technological innovation, we have provided a starting point in the field. So we have shown that it is possible to synchronize these different data streams. And from now on, since everything is open to the public on how it was built and what software is underlying it, from now on, other researchers could take this backpack and start going sensor by sensor, improving it. For example, the backpack as of course, a big computer, but in theory, you don't even need that much power. You could scale it down. Also, the EEG, you can also use, for example, different kinds depending on your question. The last event I went, I saw a new EEG innovation, which is basically recording on the ears. So they're like earbuds. They're very hidden from, you don't even know you're using an EEG. But, and it's just data from one channel. But for example, if your research question is about frequencies, it is more than enough. And one main disadvantage of these experiments was the fact that the EEG captures a lot of attention, which makes people uncomfortable. So that would be one way to improve this, going smaller EEG. Also diminishing the climate station, that might be harder, but might also be doable. And then also the backpack hides a lot of the complexity, like the cables and connections. But if in the future some research group could also diminish the amount of cables, that would be a great way to improve.
Thank you very much. We are going to the last question, but actually the question is bringing us to the beginning from Alexandra. What was the hypothesis? What did you expect actually to discover? And I guess that this is at the different levels, no? Because actually there will be different cities also studies, and it is happening in different cities, and not only from the neuroscience point of view, but also from the different other angles.
So I think it's a very fair question. So it almost looks like a disruptive question from perspective of you didn't have an hypothesis. So in a way, it is one can conceive that the hypothesis is the following: can we actually record environmental and physiological and EEG data while walking on the street in a synchronized manner? And so we tested this hypothesis. Whether this is actually going to achieve something, I think it's just what we are also trying to address now is for the time being with the data analysis to contrast this with what has been reported in the literature in other settings. But this is one hypothesis that was, if you actually want to start doing this, you have to start something. And of course we have particular questions that we are answering here, and we are not going into the detail. So we are addressing, for example, natural, more natural or less natural urban environments. So the selection of our paths are like this. So we have some other underlying more scientific hypothesis to test. But one of the key elements here was also the development of a new technology, as João was saying, in a way that was not too contrasting with what rigorous science is also about, in a sense that we have to not neglect that with one electrode we might do some, but of course it's never going to be like easy to relate with the findings from other type of more rigorous studies. But it's a very important question, and I appreciate the question.
Thank you very much. And actually, from the fMRI point of view, there is publication in Nature Cities actually that we are mentioning. Yeah. There is another line of research within this project that we can actually check already in Nature. Yes. Nature Cities. Perfect. Thank you very much. So this was the last question.
Thank you very much.
Thank you. Really appreciate your presentations, and we are expecting, waiting for the results. And thank you everyone for joining us today. And I hope you're gonna join us again for the next webinar next month. Like always, please visit our webpage neuroelectrics.com, then you will find additional information. Thank you very much, and have a great rest of the day. Thank you.
Thank you.
Thank you.
Thank you, guys. Thank you. Bye.